Good morning, everyone out in YouTube land. So, to review a bit, we're actually going to go to the figures here in the book and pick up where we left off, but this is for those of you that skipped over what I think was a brilliant lecture on Wednesday. We were talking about the mechanisms behind protein localization. The fact that the cell is not just a bag full of proteins, but in fact there's a lot of organization there. And so we've got mechanisms by which proteins can direct themselves. And it's a very interesting question that actually I was looking it up. Gunther Blobel did end up winning the Nobel Prize, I don't know, like 20 years ago. And the work that he did was in developing the understanding of the mechanisms by which cells can direct protein localization, and how a protein has an intrinsic, if you will, address or signal that says this is where I belong. And so we have two primary mechanisms by which we can localize proteins. One is after the protein's been translated, it gets targeted to the nucleus or to the mitochondria. And then we have another mechanism that is, as the protein is being translated, it's being targeted. And so we have post-translational translocation or co-translational translocation. The critical point is everybody starts out on free ribosomes. These are the ribosomes floating about the cytoplasm. All RNAs start here. Now, if the RNA has a signal sequence, the signal sequence gets translated, and that is recognized by the signal recognition particle. And the signal recognition particle, and this diagram is sort of like the first stage in Blobel's discovery. The signal recognition particle is directing the whole ribosome, RNA, little bit of protein to the surface of the ER. And that makes this become rough ER. And the protein is now being translocated through a protein channel into the ER. And so here is sort of the full refined story that Blobel and Doberstein and David Sabatini put together. And just for those of you that are paying attention, Doberstein's first name is Bernard. I didn't know that off the top of my head on Wednesday, but I do now. So, they discovered all these different parts. Here is the signal recognition particle. It is an RNA that has a set of proteins on it. And the proteins bind the signal sequence. They halt translation until this complex is able to find a ribosome, land on it at the SRP receptor. And then you have this protein channel that the protein is being slid through. And I sometimes think, I should always have like a little donut and a piece of tubing as a way of demonstrating this. But imagine you have the donut hole, you have the tubing, you have the tube land here, and now you take a loop of this spaghetti and pass it through the hole, and it slides through. That's what the co-translational translocation is doing. And so as the protein is being translated, it is actually being dropped into the ER lumen. So if we further think about this, the protein is then hung by the signal sequence. But we know there are proteins that are secreted. They're going to be dumped out. And that's where the next enzyme, signal peptidase, comes into play. It's clipping the signal sequence. So now what we've got is a protein that is floating in the ER. Our next topic is going to be what happens next by taking proteins that are in the ER and putting them into the Golgi network and allowing them to be secreted. And so that is, if you don't have any other sort of sequences of interest on the protein, that's what's going to happen. It's going to get dumped into the ER. And unless something else is on this protein, and there are certain amino acid sequences, this protein is destined to be secreted. And so it actually becomes both an interesting mechanism that's important because there's a lot of secreted proteins. It also becomes a tool by which recombinant DNA technology can build a protein that will be secreted by a plant, by an animal cell, or by a cell, a plant or animal. And from a biotechnology perspective, this is a pretty cool trick. So you can have a cell that is engineered to produce a specific protein you want, say insulin. And the protein with a signal sequence gets targeted to the ER. The ER then is going to transition into the Golgi. This protein then gets dumped out into the media. And what you can do, it's kind of the holy grail of protein production, is you have a cell system that is just constantly secreting your valuable protein. And then all you have to do is suck the media off, add fresh media, and just keep these cells rolling. And this becomes a technological way of producing proteins. So that's where the protein ends up, is in the lumen. Now, we know that not all proteins, a lot of proteins, are on the cell surface. There are membrane-bound proteins. All these different transporters, the glucose transporter, the sodium-potassium ATPase, all these guys are stuck on the cell surface. And so the next question was, how do they end up being stuck on the cell surface? And that's a good question. And it happens in that when you scan around the amino acid sequence, and this is something you can do on programs like BLAST, where, I mean, not for BLAST is searching, but you can use a computer program that will essentially scan like 10 amino acids at a time, and it will begin to characterize how hydrophobic or hydrophilic this stretch of amino acids are. And when you're doing this, you can come across these stretches, of 10 or 15 amino acids that are extremely hydrophobic. And this is a membrane spanning region, a transmembrane region. And what happens with that is the hydrophobic region will get stuck in the membrane. And so this allows us actually to begin to think about the topography or the topology of proteins as they're being stuck into these membranes. In that, with a single transmembrane pass protein, my mask is like falling on. Let me have it upside down here. Hold on a second. I have to worry about making sure my pants are zipped. And now how's my mask? Yeah, better. And my pants are zipped. Fine, I'm good. All right, so now, the protein has a stop transfer sequence of this membrane spanning region. As it's being co-translated, this membrane spanning region hangs in the membrane, and now, rather than being continued to dump through into the lumen, the translation product, the protein, is stuck out here. And so, with a single transmembrane pass protein that has a signal sequence, we can make the following observation, that the amino terminus, which is equivalent to the 5' end of the messenger RNA, is going to be in the ER lumen, and the carboxy terminus is going to be sticking out here. And when we start looking at proteins, transmembrane proteins that are single pass will generally have this architecture, which means that, we probably have a fairly good understanding of what's going on here. So it's not the only way. There are all kinds of protein stop transfer sequences that you can have, and there are, in fact, internal signal sequences that will cause this thing to get rebound, and we can build multi-pass transmembrane proteins. And so a single-pass transmembrane protein, they are around, but a lot of receptors and a lot of channels are multi-pass. They'll have like 7 transmembrane stretches. And this mechanism of co-translational translocation takes that into account and figures it out pretty well. So this is another important kind of observation and understanding. Next week we're going to be getting into hormones and hormone receptors, and it's a really interesting topic. And if we think about this, with a hormone receptor that's in the membrane, it's critical that it's arranged in the membrane properly. The hormone receptor is going to have a stretch of amino acids that can bind the hormone. The hormone is going to be extracellular. That portion of the protein has got to be facing out. And, you can't have it just pop into the membrane and, half of them are facing in and half are facing out. That would not do. A hormone receptor has got to have its receptor side sticking out in the right direction, otherwise it will not work. So these mechanisms by how proteins can embed themselves in the membrane and the topology of how they do that is in fact a pretty important kind of idea. So this is now another kind of mechanism by which we can have a double pass transmembrane protein. It gets to be quite the complicated and convoluted mechanism. The main point I would like for everybody to be able to do is describe this kind of mechanism by which, having a single signal sequence, the protein is in the lumen and is destined to be secreted, and this kind of topology where you have a single transmembrane pass. The carboxy end is going to be facing the cytoplasm. The amino end is facing the lumen of the ER. That is the critical stuff. And let's draw for a while because we can. This is what we're getting into next, is how proteins are being taken to the next step. And that involves getting proteins into the ER. And then from there, getting into the Golgi apparatus. Co-translational translocation. All right, so when we have co-translational translocation, placing a protein into the lumen of the ER, that is the inside, this is the lumen. The next step is going to be bringing this protein mix in the lumen further out. And if it's a secreted protein, that is, if it doesn't have a transmembrane sequence, it's floating around in here. And we're going to be talking about a bunch of secreted proteins in the weeks to come. So how does it get secreted? And the answer is the transmembrane system, and a big part of this is the Golgi apparatus. And I think it's actually named for like Enrico Golgi, an Italian cell biologist who discovered this in the 1800s. In plants, for whatever reason, the exact same structure is called a dictyosome. Why it's not called a Golgi, I don't know, but there you go. If anybody out there in TV land can tell me, the dictyosome. So Golgi, dictyosome, potato, it is the transmembrane system. And the ER is directly engaged and related to it in the following way. So here is the inside of a cell drawn on the board, writ large. This is going to be a very big cell. So we've got over here the nucleus. The nuclear envelope is actually a double membrane-bound sac. Each membrane surrounding the nucleus is a bilayer, but then the bilayer itself is doubled. It's a little bit like having a bunch of Ziploc bags with the film of plastic being a lipid bilayer, and you have this thing tiled in these Ziploc bags. Regardless, the ER is essentially connected and related to the nuclear envelope, but you've got these stacks of membrane here. This is the endoplasmic reticulum, the ER. Now, as we were just saying, the RNA is being transported out of the nucleus. Free ribosomes just in the cytoplasm, are going to engage, initiate translation, begin producing the protein. If the protein has a signal sequence, the ribosome is going to latch on to a spot on the ER, onto the rough ER now, and the protein gets translocated into the lumen of the ER. The next step is for vesicles to, the term is, they bleb off. That is the technical term, is blebbing. So you have a vesicle that will bleb off or pinch off from the ER, infuse its and kind of diffuse its way into the Golgi stack. My black marker is dying. The Golgi stack. Now, the Golgi is a very complicated set of membrane pancakes. The book has their analogy is a stack of pita breads. I'm down with that. If we think of pancakes, which it's breakfast time, that's what I'm thinking about. And we've got, again, a topology going on here. I'm going to see if I can't get better marker technology going here. We have these stacks of the Golgi. And they are somewhat interconnected. It turns out that things go from one side of the Golgi to the next in transition vesicles. So there's always blebbing off of membrane, little balls of vesicles that are moving from one side to the next. And so the Golgi actually has a lot of work to do, which we're going to get into in just a moment. And we can define the Golgi in a couple of ways. We've got on this side, away from the nucleus, and all right, let's go ahead and put here is our cell surface. And oh, my heavy markers are dying. How's this one? Not bad. Not great. All right. These are good, but they're a little bit skinny. Okay. We got our extracellular, or this is the external membrane of the cell. And so this is the outside world. On this side, this is known as the trans face of the Golgi, also known as the mature face. And this side is the cis side of the Golgi, also known as the forming face. And there were a number of questions back in the 70s and 80s about how the Golgi actually goes, and we're not going to get too much into the weeds, but there's some experiments and questions of, is this a set of pancakes in which the vesicle that ends up at the cell surface is being pulled off of this part of the Golgi, the trans Golgi, and making its way to the cell surface. We're going to have a vesicle fusing right there. There we go. So the question is, are the vesicles that are fusing with the cell surface coming from here, and this portion of the Golgi is essentially being dissociated, and you have new Golgi forming, which is kind of how that term came about, back here, and then you just have this stepwise, as the pancake gets eaten at the trans end, a fresh pancake is being slid up in the back. And I won't bore you with the details, but there are a number of experiments that demonstrate, no, that's not how it works. The Golgi is more or less a stationary building spot, and material moves from one side to the next through transition vesicles. So the Golgi is essentially what is known as being a stationary cisternae. The individual stacks are referred to as cisternae, C-I-S-T-E-R-N-A-E. Not that anybody cares, but a good crossword puzzle word. And the stacks are in fact stationary, and they've got specific jobs. So a couple of things that are critical happening here is a whole set of protein processing events. And a critical protein processing event that's happening in the Golgi is something that we call glycosylation. And we have talked about this a bit in our discussion of proteins. It is the production of what is known in the biz as a glycoprotein. In 2 or 3 weeks, we'll be talking about some proteins, particularly that are secreted out into the extracellular matrix, that have got so much sugar on them that they're called proteoglycans. But for the most part, membrane proteins and proteins that are being secreted are glycoproteins. And the sugar decoration is a real critical part of how the proteins work. And this is a little slightly nerd point. When you're talking recombinant DNA mechanisms by which you're going to make a protein, say you want to make something to inject into people as an immunogen for a vaccine. If you're producing a human protein in bacteria, i.e. you have transformed bacteria with a plasmid that's got a piece of the human protein. Bacteria don't do glycosylation in nearly the same way that animal cells do, that eukaryotic cells do. And so the protein is, like with insulin, it's just being produced as a raw amino acid sequence and then being dumped out. There are a lot of proteins and things, a lot of hormones that work because they're glycosylated. And if you just have just the naked raw amino acid sequence and none of the sugar decoration, it's not a functional protein. And so in order to get that, you actually have to use a eukaryotic system. And you have to have it getting into the Golgi stack. So glycosylation is a critical thing that's happening. There are a whole set of different kind of protein processing events that are happening. And this is everything from cleaving the protein, things like proteolytic processing. We've got the addition of very complex sugar chains in glycosylation. We've got sulfhydryl bonds forming. You know, we talked a little bit about this again in the case of insulin. It is produced as a single protein with a amino terminus and a carboxy terminus. And there is a cysteine, a conserved cysteine, or two in the amino acid chain. These two conserved cysteines actually form a sulfur-sulfur disulfide bond. The protein ends up folding itself up like this, where you've got the carboxy end and the amino terminus, and you make this sulfur-sulfur bond here, the disulfide bridge. Within the Golgi, there is a protein processing event that clips right there. And it makes insulin look like it's actually two proteins linked together through a disulfide bridge. And in fact, it's a single protein. And it turns out that if you don't do that processing event, insulin still works. So consequently, insulin is generated in some kind of cell batch fermentation system that they don't clip it. It still works. And so insulin is now generated from a human gene, whose gene would be actually a deeply shrouded and interesting story, because it was some dude at the technician at Genentech in 1982 that either spit into a tube or took some cells of himself and generated, in fact, this gene. They had to have actually taken a pancreatic biopsy. Regardless, the protein works, even though it's not processed. But there are a whole bunch of proteins that if they're not processed properly, they won't work. And so there are mutations that are protein processing mutations. And so all kinds of interesting things happen here. So we got protein processing, proteolytic activities, sugar being added. There's a whole bunch of protein folding events occurring here as well. So proteins are being processed post-translationally, and these are all post-translational modifications. So the protein is making its way into these different transition vesicles, making their way through the different faces of the Golgi, and sort of like an assembly line. They're being added on, and you can hear very carefully if you listen, the rivets and people zipping things off and changing structures around. The sugars get added on in a very sort of standard way. You have this long chain of sugars being attached to very specific amino acids. And then the sugars can be further processed and modified. And so you end up with some really interesting kinds of structures coming out at the end. So just as a way of thinking about the topology of our secreted protein, if we've got a protein that has a signal sequence but no transmembrane sequence, it's going to end up in the lumen of the ER. So we have co-translational translocation going on back here in the ER, and here is our protein. As it makes its way, it's going to fuse with the Golgi. It's floating here, and then it's in a transition vesicle, and it's there, and then finally it's here, and then we're going to have some of it in a vesicle here that's making its way to eventually the cell surface, and bada bing, it gets dumped out of the cell. That is the pathway of a secreted protein. There you go. So this is a critical part of how proteins are being transported and localized. And just a couple of little trivial points. It's a little bit of the, every time we're working on this, the question I think about is, who cuts the barber's hair kind of question. There are tons of proteins that are actually in the ER and in the Golgi doing all these modifications. Turns out there is an amino acid sequence that is known as the Golgi retention sequence. And in terms of trivia, why this is stuck in my head, you got me. It is the amino acid sequence YGSIR, or YGSR, the Yigser sequence. It turns out that if you've got that amino acid sequence in your protein, it will get trapped in the ER, and it won't get dumped out. So the Yigser, there you go. We have a couple of little amino acid sequences that do very critical things. Yigser is one of them. This is the ER retention signal. And so as people begin dissecting proteins and looking at their amino acid sequences, all kinds of stuff starts to fall out. We've got transmembrane regions, we've got signal sequences, we've got ER retention sequences, we have Golgi retention sequences. There's a lot going on here. And so proteins get localized in very specific ways. Now, another part of this biz, well, actually a couple of things. This side of the Golgi, known as the transface, if we were to do an antibody stain for proteins that are involved in establishing essentially roadways or the transportation systems, we'll be getting to these in 2 weeks. These are parts of the cytoskeleton, and they serve very much as railroad lines. There are tons of these lines underneath the Golgi that are stretching out. And this is all collectively known as the trans-Golgi network. So it's a little bit like a gigantic switchyard in that we've got vesicles that are flowing out. They have to get targeted to specific parts of the cell. There is a massive transit system right underneath this end of the Golgi because there's so much movement going on. The trans-Golgi network is responsible for sorting all these vesicles. And another side of this that comes about is that if we continue to take these vesicles, fuse them with the cell membrane, and dump stuff out, which you're doing right now, your cells are going to start to get bigger and bigger and bigger and bigger and bigger because you're dumping this membrane there. What we have to have is a backwards flow direction. This is known as exocytosis. We also have a whole set of vesicles forming that then pinch off, and they're moving back in. And this, of course, is known as endocytosis. So at the same rate that exocytosis is happening, we also have to have endocytosis. So exocytosis is the mechanism by which these vesicles are making their way to the cell surface and either dumping their contents, or we can have, and this is actually kind of a cool thing, let's just imagine we've got a single pass transmembrane protein. It's right here in a transition vesicle, making its way to the cell surface. If it's a single pass, that means that the amino terminus is here, the carboxy terminus is here. That's on the outside. Convenient way to think about this is the carboxy with a C in the cytoplasm. Why not? The amino terminus is inside. Oh, that's so cute. Now, when this fuses, it's going to be a little bit like a soap bubble. It's going to fuse with the cell surface. We're going to make it stretch out a little bit, like so. And once this vesicle fuses with the cell surface, we'll make a little patch of our pinky membrane, it's going to be looking like this. The carboxy terminus is facing inside still. The amino terminus is facing outside. This is a critical topological issue in that we can always remember and depend the lumen of the ER, the lumen of the Golgi, the inside of a vesicle, is contiguous, essentially, with the outside external environment. That's the face that gets stuck outside. The carboxy terminus in our single pass, or the protein part that's hanging out in the cytoplasm, always stays in the cytoplasm. This way, we can take a hormone receptor and make sure that its grabbing end is stuck out and its signaling end is stuck into the cytoplasm. A critical point indeed. So we've got exocytosis and endocytosis, and we have patches of membrane then that are being established. Another thing that's going to happen is you can have a vesicle that's going to have several proteins associated with it that need to be right next to each other. And this is a way of making sure they stay next to each other. The membrane fuses, and you've got these proteins kind of hanging next to each other. So we have a lot of organization going on here. And in terms of exocytosis, the vocabulary is pretty easy. It is just vesicles, shuttle vesicles moving their way to the surface. Coming in, we begin to bring stuff in, and there's all kinds of things that start to happen. The vesicles that are being pulled in, we're pulling in parts of the cell surface. And it's actually being pinched off and then escorted back into the cytoplasm. And we're going to go back to a couple of images here and then go back to the board, but here you go. Just a couple of definitions, so when you see them you go, oh yeah, I know what this is. Endosomes, that is, pinching a vesicle off and bringing it into a cell. That vesicle can do a couple of things. Very often, when we bring a patch of membrane into the cell, we can do a couple of things. We can just recycle the cell surface. We also maybe captured something here. As we're going to get into hormone receptors, the hormone receptor is sitting out there, it grabs the hormone. It is fairly quickly going to be endocytosed. And so the hormone is actually in here. But you can pull in all kinds of stuff this way too. This phagocytosis is this way. And so a couple of the terms is that as soon as it comes in, it's referred to as an endosome. But then the endosome can mature, and depending on things getting fused with it, it can become a lysosome, which is essentially a trash can. Lysosomes are carrying stuff that was on the cell surface, and you maybe pick something up, maybe you're just recycling the cell surface. You can have a whole set of enzymes getting dumped into that typically have a low pH activation point. And there, in fact, is going to be a proton pump engaging in this endosome start putting protons into the endosome, dropping the pH, and these low pH activated hydrolases turn on, acid hydrolases. And one of the ideas that's brought forward is you can have enzymes that break down stuff and allow it to be recycled. Or they're breaking apart cholesterol that you brought in, and you're going to recycle the carbons and things. The enzymes, the hydrolytic enzymes, are activated at low pH that is happening in this vesicle. That way they're not chewing around in the cytoplasm. And so it's actually a rather clever little technique there. All right. Let's see. We have a story here. Let's go for a moment. And yeah, so here is an example of an asparagine-linked sugar being added in the ER lumen. Proteins being folded by chaperones. We talked a little bit about anything right here. Here is our Golgi stack. So we've got the cis Golgi next to the nucleus, the medial cisternae, and the trans Golgi network. And then here is a stack of pita breads, and I assume the olives are transition vesicles. That makes sense to me. And that would make the pimento the secreted protein. You can never go wrong by taking an analogy too far. Let's see, anything good here? This is Global and Doberstein's experiment. Fine. All right. Our last little bit here is we know how the vesicles are being formed. And this was a discovery back in the late 70s, early 80s, looking at membrane of highly secreted, the big-time secretory cells that do a lot of secretion. And something people began to note was a patch in the membrane that had this little fluffy area on it, and it was known as a coated pit. And so you have this, dimple area that has this fluff. This is a coated pit. Now, this is one of the examples of where the static nature of electron microscopy requires some creativity, because what people are calling a coated pit was in fact a vesicle on the way to being formed, and they didn't know it. So we've got the coated pit is actually being pulled in, and there is a protein that surrounds the vesicle as it's being pulled in, or this portion of the membrane, that causes it to be pinched off. And that's where we're going to go back to the board. So part of this is there's a lot that we know about this mechanism. And as I'm sitting here thinking, we could have just moved on, but there's a delightful Star Trek story that I have to throw out. So there. And when I get to it, you're going to absolutely go, oh my God, you spent close on to 10 minutes of my life just to get to this Star Trek point? Yes, I did. Stop. So, we've got membrane. Let's go now and draw us a little bit of cell surface here. Here is our cell surface. This is the outside, or the extracellular space. And this is the cytoplasm. And we're going to have a patch of membrane that gets pulled in. And it's a way of forming a vesicle. And why I was saying that, with a static electron microscope, you have to have a little bit of creativity, because we would see a fluffy little area happening, and then that fluffy area starts to pull in, and then it pulls in some more, until finally we've got a vesicle. And that is, in fact, what's happening. There is a protein that lands on the inside of the membrane and causes the membrane to get pinched or puckered, pulled it out, pinch it off as a free vesicle. One of the key proteins in this coating is a protein called clathrin, which would be an interesting name for your daughter, but maybe a better name for your cat. Clathrin is a protein that begins to form on the portion of the membrane that's going to get pinched in. And it forms a structure like this. Let's see if I can draw it. Like that. And it is known as a triskelion. So the clathrin protein is able to autocatalytically form up and actually begin to link together and apply a torsional force on the membrane to begin pulling it in. Think about taking a sheet of plastic or like a sheet of rubber that you can begin to pucker and pull. And what clathrin is doing is linking itself around it and actually causing the vesicle to form. The Star Trek point, and I knew you were waiting for it, is there is a original series Star Trek episode called The Gamesters of Triskelion, in which the crew, in some kind of transporter accident, is beamed a million miles away, and they end up on this pad that is shaped like this. And this is the Gamesters of Triskelion have hijacked people from around the solar system or the universe, the galaxy, to participate in these cruel games. And they've got some sort of neck thing. You've seen this?
I've seen all of it, yeah.
Oh, good. Okay, and I really almost want to turn around and say, see, Hannah has, the Gamesters of Triskelion. And the story is either that the guys that discovered this structure actually had been watching Star Trek and said, hey, that looks exactly like the Triskelion on Star Trek. So I think Star Trek had the term first, and then clathrin has it later. Yeah, see, I told you. I had to go through the clathrin just for the Star Trek reference. Thank you very much. Now, this is a way we can autocatalytically begin pinching these proteins off. And so the vesicle looks a lot like a Chex cereal cube. I don't really think I'm all that hungry, but we have pancakes, we have Chex cereal, we have pita breads and olives. We're about to stop because I have to eat something. So here is the coated pit. It gets pinched in, and you make a little vesicle. So it's a little bit like if you've ever played with a camping tent, in which you've got the poles that you have the tent as a flat surface. You start sticking those poles in the sleeves, that is essentially the clathrin, and you can begin bending it, and you make a three-dimensional structure. So there's our little Chex cereal blob, and this is clathrin forming. So you have the extracellular space. Clathrin forms up around this patch of the membrane, begins to autocatalytically pull itself in, pinches the membrane off, and then it can be uncoated, and then you've got a naked transport vesicle that can go on and do things. So there are a whole range of coated vesicles. We need not worry about them, but there you go. There is, in fact, a whole system involved in targeting these different vesicles as well. Turns out that one of the first identifications of what are known as snares, then there's the T snare, which is on the target, and the V snare on the vesicle, are responsible for specifically bringing vesicles to certain spots. It was discovered in a fly mutation that was done with a screen looking for neurogenic defects, in that the synaptic vesicles that we talked about during neurotransmitter operation. The synaptic vesicles have to target that end of the axon so they can dump the neurotransmitters into the synaptic cleft. And, you'd be a really bad synaptic or a neurotransmitter-filled vesicle if you went the wrong way. It's like, this poor neurotransmitter vesicle, he's going over here and he's going to fuse back here. Like, dude, there's no synapse over there. Everybody go over here. And the T snares and B snares are responsible for that. Just thought I'd throw that out there. And so we talked a little bit about protein folding, just to throw this out as well. The ER is the place where protein folding happens. Protein folding is becoming an increasingly critical idea in terms of how proteins work. That misfolded proteins are bad news. Misfolded proteins are responsible for mad cow disease. Misfolded proteins are responsible for Huntington's disease. Misfolded proteins are probably responsible for Alzheimer's disease. More and more information about protein folding is critical. Just throwing that out there. And these proteins that we used to call heat shock proteins, now typically referred to as chaperones, they're responsible to help fold proteins. So you have the properly folded protein and the improperly folded protein. And that's about all we have to say. Pita breads, Global and Doverstein. So this is a good kind of general purpose review diagram. We had the Golgi apparatus, the trans-Golgi network. The vesicles are being pulled off, blebbed off. Here's a secretory vesicle. It's being bound to or fusing with the cell surface. This is a form of regulated secretion. We then have receptors that can bind signals like hormones, different things, and they can get pulled back in. We also have lots of things like new plasma membrane that is just really kind of constantly going to the cell surface. This is unregulated and constitutive secretion. You got to balance them out, otherwise your membranes get flabby, though. So another sort of diagram that if you can explain that, you're doing good. And here is a bacterium being engulfed by a phagocyte. Yeah. We'll talk a little bit about this when we get into receptors and things. The mechanism by which receptors can be pulled from the cell surface, recycled, and put back out was first discovered with cholesterol metabolism, but we'll get into that later. And nothing all that exciting here. So this is the acid hydrolases. The fact that vesicles can have the pH drop pretty good, significantly from 7.2 down to 5, and that activates all these different hydrolases and things. And I'm not sure what this is trying to just show us, but there you go. All right, so that is secretion and protein processing, protein processing. What we're going to next turn our attention to is potentially one of the most important topics in molecular cell biology, and that is cell signaling, the way hormones work. And it's a really important topic, and it uses the cell surface. And then after that, we're going to talk a little bit about the cytoskeleton, some of the components of the cell that give your cell rigidity, act as transport mechanisms like the roads and the ability to contract and expand cells. And then we will finish up on our last week of lecture talking about the extracellular matrix and tying cancer biology into all of this stuff. And so that's where we're going. And that's all I have to say about that. So, bye-bye.